Bismillah ar-Rahman ar-Rahim. This is an overview about the different types of biochemical tests which we have discussed in the overall 3 lectures. So, in the first lecture we discussed about the bile solubility, catalase and coagulase test. Bile solubility is basically to differentiate the different types of gram positive cocci, the strep pneumoniae, from the other types of streptococci, and this is basically performed on a agar which to which the bile salt sodium deoxycholide is added, and these are the requirements which is needed for the tests, and these tests can be performed on the slide and whereas on the tube and directly on the culture plates. So, the procedure is written here, that first take the physiological saline and then emulsify the colonies in that and make turbid suspension, then divide into two, add the bile salt into one tube and then the second one is left alone, or normal simply water is added for negative control. Then after 10 to 15 minutes and 37 degree centigrade incubation, then the clearing of the turbidity is checked. So, this is the then results that the test organism which clears the turbidity, that is probably Streptococcus pneumoniae, the gram positive cocci and in chains, and the one who do not clear the turbidity, that is other than the Streptococcus pneumoniae. So, this is the controls. Every test which is shown here have their purpose. This is the purpose of the Catalase test, to differentiate between the different types of gram positive cocci, that is in chain, and gram positive cocci, that is in the clusters form, in the grapes form, especially staphylococci, to differentiate staphylococci from the streptococci, because staphylococci, they are catalase producing and they break down the hydrogen peroxide into oxygen and water, and this test is also performed on a slide as well as on the tube method. So, these are the results of the Catalase test, in which active bubbling is produced after adding the growth into the hydrogen peroxide solution, and it gives bubble, and the controls are Staphylococcus species is the positive control, whereas Streptococcus species is the negative control. This is both the slide test and the tube method of the Catalase test. These are the cautions which is required during the test. The third and important test for the differentiation among the different types of staphylococci, the Staphylococcus aureus, Staphylococcus epidermidis and the saprophyticus. So, the coagulase test, coagulase enzyme is basically produced by Staphylococcus aureus, and it is an important which clots the blood. This is the method of performing the Staphylococcus aureus producing coagulase test, and the plasma is added to this test, and the result shows on the slide that when clumping occurs within 10 seconds. So, this is basically a Staphylococcus aureus species, and if there is no clumps, there is no clot. So, this is not a Staphylococcus aureus. On the slide method, bound coagulase is basically determined, and on a tube test a free coagulase is determined, and this is the procedure of the tube method. Then the results of the tube method, if the clotting occurs within the specific time, and so the Staphylococcus aureus makes clot of the fibrin, and if a clot is not produced, then this is not a Staphylococcus aureus. This is the picture of the test tube method for performing the coagulase test. Here is the clot formed, and this is the negative control, the negative test where the clot is not formed. In the second lecture of these biochemical tests, we discuss about the other 3-4 tests, the citrate test, the DNase test, indole, and the litmus milk decolorization. So the citrate utilization test is needed for the identification and differentiation among the different types of the enterobacteria, and it is performed on cymate citrate agar as well, and also performed with a different methods of tablets. So in cymate citrate agar, they gives us a blue result, bright blue color from green to blue, and this shows us a positive citrate, and when there is no change in color, then there is no citrate positive, it is negative. So this is the example of a citrate negative test, and it is required for certain enterobacteria. The citrate utilization test using the tablet procedure, and in this the citrate tablet is added to the saline solution. This also shows us a red color of the medium or the solution when citrate is positive, and if the other color, yellow or orange color, is produced, then the citrate test is negative. The DNase test is an alternative test if we don't have the plasma for performing the coagulase test, or if there is any issue with the coagulase test interpretation. So this is also an alternative for that test, and it is based on the organisms they produce DNase. So the organism is cultured in a media which contain DNase, and after that a weak acid is hydrochloric acid is added and poured to this, so which clears and hydrolyzes the DNase. So the positive result gives clear areas around the colony. So this shows us that the DNase is positive, and which do not clears the area around the species around the colony. So that is the DNase negative, and the control for DNase positive are the Staphylococcus aureus, and DNase negative control is the epidermidase. This is the pictures which shows this is the positive test 1, 2, 3 and 4, and the other is a negative. This is another example of the DNase tests. The indole test is also required for the identification and the differentiation of different enterobacteria, E. coli, Proteus vulgaris, Redgeri, and Moraxella morgani and Providentia species. They produce indole, and it is also performed in a tryptophan and in different ways. So the first, it can be used as a tryptone water with coax reagent, then the beta glucuronidase indole test tablet, and the third is the lysine decarboxylase indole tablets. So the three methods is discussed here for performing this test. This is the procedure of the indole test. This is the result of the indole. If this surface layer produces red color ring, then this is indole positive, and then surface layer do not produce red color ring, then it is negative. So this is the picture of indole positive and negative. Then using beta glucuronase tablets, and this is the result of the test of the beta glucuronidase tablets, and if yellow color is produced, then it is positive for PGUA, and if there is colorless, so this is negative. And then coax is added, which gives red surface layer or red or pinkish surface layer, and if yellow surface layer, then it is indole negative. And this LDC lysine decarboxylase also is performed, and it is a tablet mixed with the indole. So the positive LDC gives a blue or violet color, and negative LDC gives a yellow, green or gray color, and this is the result of the end result of the indole LDC tablet result. These are the tablets which are required for performing this indole and LDC and beta glucuronidase tablets. The litmus milk decolorization is also an inexpensive and alternative for the identification of different types of enterococci, and the strains they decolorizes the litmus milk by the reduction, and this is the principle and the method of performing litmus milk decolorization. These are the results when a white or pale yellow pink colonies appear. So, this is the Enterococcus species, and when no change or pink color appears, so probably this is not an Enterococcus. So, the controls are Enterococcus positive control and Viridin streptococci is negative control. And another test, esqualene hydrolysis, is also used as an alternative to the bile in esqualene agar, which is basically very costly, and this is a cost effective and cheap test. Easily identifiable organisms can be identified, enterococci especially, on by performing this test. In the third lecture of this biochemical test, we discussed about the cytochrome oxidase test or the urease test and the triple sugar iron test. The purpose of cytochrome oxidase, the organism which produces oxidase. So, this is the method and the results when a blue or purple color appear after applying the Coagulase to a filter paper having the cytochrome oxidase solution, then it gives us positive oxidase test, and if no color appear or late appeared, then this is negative oxidase test. The controls for the oxidase is the Pseudomonas aeruginosa, the positive control, and the E. coli, it is negative control. And these are the cautions which is important during performing the oxidase tests. And another method of performing oxidase is the reagent strip method of oxidase, and which gives red purple color. This is the results of the oxidase positive, and this is the oxidase negative. The urease test very important for the differentiation of different types of enterobacteria in which the protease is a strong urease producer, as well as the different types of bacteria, especially the bacteria which causes infection in the stomach. This is the principle of performing urease test, and the two methods, one is with the tablet method and the other is with this is the results of the tablet method and the result of the Christensen modified urea broth. So, this is the procedure, and the result that pink color gives us, shows us that this is a positive urease producer, and if there is no pink color, this is negative. So, the E. coli is negative urease producer and the Proteus vulgaris is a positive urease producer. The TSI, very important test in the differentiation and identification of different gram negative bacteria, especially the related to the enteric tract, and there is a slight differentiation among the TSI and the KIA. The KIA contain glucose and lactose, whereas the TSI contain glucose and lactose along with sucrose, and the iron along with the hydrogen sulfide producer and oxygen gas producer is also detected in this test. This is the procedure for performing the biochemical test of TSI. First, the bud is inoculated with the straight wire, and then the slant is inoculated in a zigzag pattern and incubated for 24 hours. This is the reaction of TSI for different types of known enterobacteria and their acidic, their slant, their bud reaction, the gas production and the H2S production, and these are the different controls for the TSI. So, that was all for the overview of the different biochemical tests which we have discussed in the all three lectures about these. Thank you.